I talk about home use of an artificial beta cell in type 1 diabetes. I study in nursing school San Vicente of Paul. My name is Joscelyne and my teacher is Moscoso. Definition. It is a lifelong disease chronic in which there is high level of sugar glucose in the blood.
Type 1 diabetes is a condition in which your pancreas does not produce insulin, a hormone your body needs to maintain proper blood sugar levels. After you eat foods that contain carbohydrates, chemicals in your small intestine break them down into single sugar molecules called glucose. Next, the cells lining your small intestine absorb the glucose, which passes into the bloodstream. When the blood reaches your pancreas, beta cells inside the pancreas detect the rising glucose levels. The beta cells release insulin into your bloodstream to reduce glucose levels and to keep your blood glucose in a healthy range. Most cells of the body have certain receptors on their surface that bind to the circulating insulin. Insulin acts like a key in a lock to open up the cell so that the circulating glucose can get inside the cell. Now your cell can use the glucose to produce the energy it needs to function properly. If you have type 1 diabetes, your pancreatic beta cells lose their ability to produce insulin, resulting in high blood glucose levels and other complications. In type 1 diabetes, your immune system, specifically your white blood cells, mistake your pancreatic beta cells for foreign invaders. In an autoimmune response, your white blood cells secrete autoantibodies that destroy your own beta cells. As a result, your pancreas produces little or no insulin. Without insulin, glucose cannot get into your cells, so they are starved for the calories they should be receiving from glucose. In addition, the glucose level builds up in your bloodstream, resulting in a condition called hyperglycemia. Common symptoms of hyperglycemia in type 1 diabetes include excessive hunger, excessive thirst, frequent urination, unexplained weight loss, fatigue, weakness, irritability, and blurry vision. If hyperglycemia is not treated, you can become severely ill. Because you don't have enough insulin circulating in your blood, your cells can't use glucose for energy. As a result, your body breaks down your fat and protein stores as an alternative source of energy. As fat breakdown continues, certain byproducts, known as ketone bodies, accumulate in the blood, resulting in a condition called ketosis. When ketones build up to dangerously high levels, a life-threatening condition called diabetic ketoacidosis results. If your blood glucose remains high over time, long-term health problems can occur, such as atherosclerosis, blindness, nerve damage, and kidney disease. If you have type 1 diabetes, your goal is to keep your blood glucose within a normal range. This is done through a combination of proper insulin replacement, monitoring your blood glucose, and, just as importantly, eating a healthy diet and getting regular exercise. Because your pancreas no longer produces insulin, you will need to take insulin to replace what your body should be making. A licensed healthcare professional can train you to inject the insulin just under the skin. You will need to give yourself injections several times each day and rotate injection sites to avoid tissue damage and absorption problems. Another way to get insulin is through an insulin pump, which is attached to your body and delivers insulin through a tube implanted just under your skin. You will need to check the level of glucose in your blood several times a day with a glucometer. To do this, you will prick your finger with a small needle called a lancet and place a drop of blood in the glucometer. Knowledge of your blood glucose level allows you to adjust your insulin dose, calories you eat during meals, and physical activity. You will need to eat a healthy diet and get regular exercise to manage your glucose level and reduce your risk of cardiovascular disease.
Application in medicine. A change has been to get the stem cells to become authentic and functional pancreatic cell, especially the beta cell that secretes insulin, as has not been achieved. The current method is immature pancreatic cell growth and wait for the body to do the work of transforming them into beta cell. Pro. Automatically control the glucose level, the blood substitute with providing an endocrine function of a healthy pancreas.
Beta cell creation may get a boost from a new technique developed over the last decade. Scientists could take skin cells, or any other cells of the body, and make them into something new, including beta cells. This process, being probed in the lab of Dr. Rohit Kulkarni, holds promise for the damaged cells of people with diabetes.
You can take a cell from an individual, maintain the genetic makeup of that individual, and use that cell to generate adult cell types, which would otherwise not have been possible.
Induced pluripotent stem cells, or iPS for short, have the remarkable power to become anything in the human body. Unlike the controversial embryonic stem cells, iPS cells come from an adult. They once lived day to day as, say, any normal skin cell would, until one day they get picked and brought into a lab. After some tinkering, they revert back to their pre-skin cell state, back to the stem cells, with the potential to become any cell in the body. In 2006, a Japanese scientist named Shinya Yamanaka figured out how to force this reversion to stem cells in a lab, using 4 specific proteins, now known as the Yamanaka factors.
We can get these stem cells starting with a skin biopsy, so you can just take a small amount of skin from a living patient, or you can take a blood sample.
Then, researchers bathe their harvest in a solution of the Yamanaka factors for about a month until enough iPS cells emerge. Once in their stem state, researchers can begin to push them forward again into whatever cells they need, beta cells in the case of Dr. Kulkarni's lab at Joscelyne. They have cells from people with type 1 and type 2 diabetes in the works, and have already published research on MODY, a type of diabetes caused by a single gene mutation.
What we have done is we have recruited patients from the same family. So one person who is not diabetic, one person who has a mutation, the MODY mutation but no diabetes, and another subject with the mutation and developed diabetes. So we can derive iPS cells from these patients with MODY, differentiate them in vitro, and understand what has gone wrong when their pancreas develops and what really causes their beta cell dysfunction.
By monitoring the development of these cells from 3 different people, they can understand how the disease progresses, and they are looking into the possibility of fixing the MODY mutated gene in the lab and then putting the corrected cell back into the subject. Dr. Kulkarni's lab currently has cell collections from dozens of individuals with different types of diabetes.
So for type 1 and type 2 diabetic patients, we want to differentiate these iPS cells towards pancreatic beta cells and compare them with iPS cells and differentiated cells derived from normal subjects. So by comparing these two types of beta cells that are derived in vitro, it will help us understand the molecular mechanisms underlying some of these beta cells that are dysfunctional in type 1 and type 2 diabetic patients. What we want to do is to derive sufficient numbers of these beta cells and then subject them to different kinds of drugs and chemical compounds to see, for example, if we can induce more beta cell proliferation.
Dr. Kulkarni's lab isn't alone at Joscelyne. Other labs study new ways to treat complications with iPS cells by trying to create liver, kidney, heart, and nervous system cells out of skin cells.
These cell types in living patients are not accessible for us to study. So one cannot go and take a small part of the heart cell or a nervous system, but the iPS allows this approach to make it a very unique resource, which would otherwise not be possible.
Each cell holds the genetic code of the person it was taken from, so this approach can allow for personalized medicine by screening each individual's cells to see which intervention method they'll best respond to.
The long-term goal would be to create large numbers of these cell types at the bench and put it back into the patient where exactly the disease is occurring. So for example, in patients with diabetes, we want to have a large bank of beta cells which are derived from this patient's cells, and then we want to put it back into the pancreas where they belong.
Because these new beta cells would come from the patient, transplantation of these cells could avoid the host rejection issues inherent in other beta cell sources. More labs at Joscelyne are looking at treatment of obesity, insulin resistance, and skeletal muscle dystrophy with iPS cell techniques.
The iPS field is moving very fast, and a lot of exciting work is progressing at the moment.
So the Joscelyne has made a major initiative by undertaking at a very early stage the iPS cell approach, which I think is going to keep them in good stead over the next 10 or 20 years, because that's where the advances are occurring.
Cons. The downside to this type of artificial cell is that no one is sure how long the implant cells survive.
So what happens when blood sugar gets too low is that patients can slip into coma or even die. Patients with diabetes are constantly trying to walk a tightrope along high blood sugar and low blood sugar. And they have to do this for the course of their entire life. ViaCyte's approach is to perform cell therapy for diabetes. The source of the cells that we'll deliver come from embryonic stem cells. We use a series of chemicals to change these stem cells, and ultimately the cells transform into cell types of the pancreas. These cells of the pancreas are then delivered to patients inside of a capsule.
Issue in Ecuador. In our country has not yet implemented this type of method to replace the pancreas by beta cell. This process is called artificial pancreas. It is developed very common in countries like the United States and the United Kingdom.
New findings about the role of pancreatic beta cells are changing our understanding of type 2 diabetes and improving our ability to treat patients. The pathogenesis of type 2 diabetes is characterized by declining beta cell function in the presence of rising blood glucose levels and relatively constant insulin resistance. In response to insulin resistance, normal healthy beta cells will compensate with increased insulin production, and the patient will remain euglycemic. In contrast, susceptible beta cells cannot compensate, failing over time to produce enough insulin to meet demands, and eventually leading to prediabetes and the vicious cycle of hyperglycemia. Although the process of beta cell decline is not fully understood, mounting research suggests that genetic mutation may underlie beta cell susceptibility. Underlying susceptibility in a subset of the population may help explain why only approximately 1/3 of obese people go on to develop diabetes. Only people with susceptible beta cells are unable to compensate for stressors that normal beta cells are able to withstand. Recent studies suggest that the process of beta cell dysfunction begins early in the natural history of the disease. As insulin resistance persists, beta cell function decreases up to a compensatory threshold that represents the beginning of symptomatic diabetes. After this point, beta cell dysfunction accelerates rapidly, and insulin secretory capacity may be permanently lost. Checking glucose values is the best way to determine if beta cell decline is beginning, especially for patients who may not yet display overt signs of diabetes but may have a family history of type 2 diabetes or other risk factors. Rising glucose values are a sign that beta cells are no longer able to supply enough insulin to meet the body's demands. In the early stages of beta cell dysfunction, the disease process is potentially reversible. If patients are treated early and aggressively with a combination of diet, exercise, and/or medications that promote a more normal relationship between beta cell function and peripheral insulin sensitivity, we may be able to delay, prevent, and even reverse the illness. Studies increasingly suggest that addressing beta cell dysfunction before insulin secretory capacity is permanently lost may promote safer, more effective care for this vulnerable population. The Beta Cells in Diabetes website is intended to help healthcare providers better understand beta cell physiology and the clinical implications of new research. Many viewpoints, levels of depth, and formats are presented. We invite you to explore, discuss, and share this information.
Conclusion. An artificial pancreas known as a closed-loop system because it requires user feedback could solve problems in taking care of the process of making decisions and decide how much insulin is needed at any given time.
When our body is working normally, the foods we eat, containing proteins, fats, and carbohydrates, are broken down into simpler, easily absorbed nutrients. One of the carbohydrates is a form of simple sugar called glucose. It is the main source of energy for the body's cells. Glucose circulates in the blood where it is available for body cells to use. Glucose is the main source of fuel for muscles, heart, brain, and other important organs. Insulin helps cells take in glucose and convert it to energy. Insulin is secreted by the pancreas. After it is secreted, insulin attaches itself to cells throughout the body at places called insulin receptors. Once attached, insulin acts like a key on the cell membrane, allowing sugar or glucose from the food we eat to enter the liver, fat, and muscle cells. The glucose in the bloodstream is decreased, and once the glucose is inside the cell, it becomes a source of energy.
Thank you for your attention.